Hello everyone. This is Anum Khalid. I am a PhD student and affiliated with the School of Life Sciences, Shanghai Ocean University, and School of Life Sciences, Anhui Agricultural University, China. Today, the topic of my presentation is effect of different concentrations of water-soluble astaxanthin on the antioxidant activity of Hep G2 cells. So first we need to know what is astaxanthin. Astaxanthin is a pigment that belongs to the carotenoid family, and it belongs to the xanthophylls within the carotenoid family, because xanthophylls are the oxygenated derivatives of carotenoids. It is a novel carotenoid with the high market value in pharmaceutical as well as feed industries, and this compound is ubiquitous in nature, especially in the marine environment, and is probably best known for electing the pinkish red hue. As you can see in the picture, this compound has beautiful red color, and in powder form it gives the appearance of velvet. And the more interesting about this compound is that it is known as the king of carotenoids because of its highest antioxidant activity among the carotenoids. And because of this reason, this compound is a part of many cosmetic almost every cosmetic product. So this compound is just love for girls. So next we see the structure of astaxanthin. This is the chemical structure of astaxanthin. Sorry. Astaxanthin consists of two terminal rings which are joined by a long polyene chain. And this molecule has two asymmetric carbons located at the 3 3 position of the beta ion 1 ring with the hydroxyl group on either end of the molecule. And in case if one hydroxyl group reacts with the fatty acids, it form monoesters, and if both the hydroxyl groups react with the fatty acid, it form diesters. And the red color of this compound is due to this long chain and conjugated double bonds at the center of these two rings. So next, what are the aims of this study? The study aimed to determine the optimum culture and growth concentration of water soluble astaxanthin for Hep G2 cells, and to determine the cell viability and morphology of Hep G2 cells, to determine the antioxidant activity of Hep G2 cells by using different assay kits, and to determine the effect of water soluble astaxanthin on the reactive oxygen species in the Hep G2 cells. So next, if we look at material and methods, first we need to get the cells. We get Hep G2 cells from the stem cell bank of Chinese Academy of Sciences, and we cultured these cells in a culture medium MEM and FBS with the 10% of penicillin, and the culture bottles were maintained in a humidified incubator with the 37 degrees centigrade temperature and 5% of carbon dioxide concentration. Incubation time was 24 hours, and then the cell were dissociated by using trypsin, which is a dissociating agent. Next, we used different dose concentrations of water soluble astaxanthin, such as 0 for blank, 1, 10, 100, 250, 500, and 1000 microgram per ml for 24 hours. Next are the cell viability assay. So to check the cell viability, we used MTT assay, and to determine the morphology of cells, we checked the cells under electronic microscope. So we determined the cell viability by MTT assay using a kit, and we followed the manufacturer's instructions. Next, we determined the antioxidant activity of Hep G2 cells grown in different concentrations of astaxanthin, and we used intracellular catalase, total superoxide dismutase, and we determined melanodialdehyde content and glutathione peroxidase activities, and also we determined all these. Enzyme activities by using different kits according to the manufacturer's instructions. Further, we determined the reactive oxygen species, and for this, we use a redox sensitive fluorescent 2,7 dichlorofluorescein diacetate, which is known as DCFHDA probe. Next comes the results. So the results showed that water soluble astaxanthin improved the antioxidant enzyme system of Hep G2 cells at the level of 500 microgram per ml. And furthermore, astaxanthin quenched the reactive oxygen species in dose dependent manner, which means that it start increasing from 1 and start increasing towards the 1000, and it was found maximum at 1000 microgram per ml. However, the study was examined the antioxidant behavior of astaxanthin in only cultured cells. So, to implement this study for human health, we need further studies. And that's about all about my presentation. If you have any question, you can ask. Thank you for your attention. Thank you so much. Bye bye.